you're looking at uh, the portfolio quiz for standard 3.2. This is part one. I'll read the directions. Label the diagram with the following terms: DNA polymerase, new strand, nitrogenous bases, original strand, and replication fork. Here's the diagram. And on this is page 63 of your reading of the textbook, you can find a picture that exactly matches the picture in the quiz. So you can just use that picture to answer questions 1 through 5. For questions 6 through 10, you're going to read pages 62 to 64. You can also watch this video here, um, and I'm going to post some videos right here in different languages that explain how the DNA replication process works. Um, they'll be on the left side of the screen here. So you can just click on it and go to whatever language you're most comfortable uh, having this explained in. Let's read questions 6 through 10. 6. Explain the role of DNA polymerase in replication. Number 7. How is the complementarity of nucleotides important in replication? Uh, that just means, like, why are certain bases, why does one base only match up with a very specific base on the other side of this double helix? What would happen if this complementarity did not exist and nucleotides paired up randomly instead? 8. Briefly describe what happens during replication. Uh, Question 9. What occurs at the replication fork? And 10. How does replication in eukaryotes differ from replication in prokaryotes? Okay, let's answer number 6. If we turn to page 63, and you look at this picture right here, you can see this bubble. That's DNA polymerase. That's an enzyme. And what it's doing is it's taking uh, individual nucleotides like this and joining them onto these incomplete strands right here. Uh, the word polymer means a combination of things. What DNA polymerase does is combine things to make new DNA. I'm sorry, not make new DNA, to copy this original strand right here. It's adding bases back onto this new strand right here. Number 7, how is the complementarity of nucleotides important in replication? Now, if we look here on page 62, there are 4 bases. There's adenine, thymine, guanine, and cytosine. And each one pairs up with will only pair up with another one of those 4. So adenine A always pairs with thymine T, and guanine always pairs with cytosine C. So always remember that A and T go together, and G and C go together. Uh, Now think about what DNA, for this second part, what would happen if this complementarity did not exist and nucleotides paired up randomly. Try to remember what DNA is and why your body spends so much energy trying to preserve the integrity or copy as your DNA as closely as possible. Um, what kind of genetic problems would happen if your DNA was not copied correctly? Question 8. Briefly describe what happens during replication. If we turn to page 62, you have a description of what happens in the replication process here and here. Now the answer to that will also give you the answer. That part will also give you the answer to question number 9. Question number 10, how is replication in eukaryotes different from replication in prokaryotes? Remember, replication is to copy the DNA. On page 64, this whole page will explain what the difference between prokaryotes and eukaryotes are, at least as far as copying DNA. In prokaryotes, replication is mostly in a loop. In eukaryotes, replication will happen at more than one point. I'm sorry, replication will not happen. It will happen more in a double helix. Also, the prokaryotic DNA, or copying the prokaryotic DNA, is a lot quicker. There's a lot less DNA, and it's copied with a lot less accuracy. So there are a lot more mistakes in DNA copying with a prokaryotic organism. Okay, now you're looking at part 2, RNA synthesis and coding. I'll read the directions. Label the diagram with the following terms: cytoplasm, DNA, nucleus, RNA, and RNA polymerase. Then answer the questions below. This will not be found directly in your reading, but you can use some of the stuff, or we will find readings for this in class. But question 6 says, I'm sorry, and you also have the videos on the side here, which can help you answer these questions. What is the role of RNA polymerase? How is it different from the role of DNA polymerase? For questions 7 through 12, use the diagram to the right to find the amino acid encoded by each codon. So what you're going to do is read these 3 letters and find the amino acid, and the amino acids are around this circle that matches these codons. So you have guanine, uracil, cytosine. Where would you find that combination, and what kind of amino acid will it make? All right, let's go to part 3. All right, now this is part 3. It says choose 2 questions. You only really need to choose 1 question. Make sure you write this on a separate sheet of paper and indicate what question you're answering. Again, I'll have the websites on the side here that you can use to answer each question. Okay, number 1. In DNA, a sequence of 3 bases is a code for the placement of a certain amino acid in a protein chain. The table below shows 8 amino acids with their abbreviations and DNA codes. Question, Part A. Which amino acid chain would be produced by the following DNA base sequence? CAA, GTT, AAA, TTA, TTG, TGA. For this, you're going to read each 3 letters and match them to an amino acid on the side here. So CAA is val for valine. For every 3, you're going to write the amino acid below. So GTT is glutamine, so you would have valine for CAA and glutamine for GTT, and you'd have an amino acid chain underneath of that, and you would just write that right there. B. Describe how a protein would be changed if a base sequence mutated from GGA to TGA. You're going to write a new amino acid sequence and say which amino acid is different between the 2 sequences because of a 1 letter change here. Or you could look at this chart and say, well, GGA is proline, TGA is 309. So what would happen to this amino acid chain? What protein would be different? Number 2, the amoeba is a single-celled organism that reproduces asexually by mitosis. Explain why all the offspring of a single amoeba can be considered clones. Remember, amoebas don't reproduce sexually. They make copies of themselves. So if they're making copies of themselves, every amoeba has the same DNA. If things have the same DNA, like identical twins, they're going to be very close to one another. That's what you want to explain in this answer for this question. Number three, identify one human genetic disorder that is caused by the inheritance of an abnormal number of chromosomes. What effect does this condition have on the offspring? Um, there are many genetic diseases you can choose from here: Huntington's, Down syndrome. Just identify, just name it, and give what some of the symptoms of that disease are. 4. How might the techniques of genetic engineering make it possible to treat some diseases caused by genetic disorders? Provide an example. So again, think about those diseases and think about um, what genetic engineering is. You're changing the DNA of something. Uh, how would changing the DNA of this person uh, not yet born prevent them from having this genetic disease? And genetic diseases are things that disorders caused by um, problems with that person's DNA. Okay, uh, if you need more information or you need more help, you can always go to our website, jvsadp.edublogs.org. For this standard, you want to click on 3.1 through 3.6, and we have a lot of websites, our Quizlet decks to help you um, with this particular quiz. All right, thanks a lot.